Hi, this is Ling from 3D Science Modeling. Both incomplete dominance and multiple alleles may cause multiple traits. How can students use evidence to differentiate these two situations? Today, let's talk about a lesson in which students used computer evidence to distinguish incomplete dominance from multiple alleles. It can be used to extend your students' understanding of incomplete dominance through data analysis and interpretation. If you have not seen my previous video about how to identify multiple alleles, you may find the link at the top. Incomplete dominance is a condition in which a heterozygous individual shows a phenotype intermediate between the phenotypes of two homozygous traits. In general, students may gain the basic idea of incomplete dominance quickly based on some conspicuous intermediate traits, such as semi-curly hair or pink flowers. What if they come across a case in which a new trait does not look like a diluted form of the parents? How do they figure out whether the trait is caused by incomplete dominance or a new allele? Let's continue exploring the inheritance in common Australian lady beetles to find out. In class, we first quickly review cases 1 and 2 with students. The lady beetles display two traits in case 1. Computer data support a two-allele model where the nice-spotted allele is fully dominant over the black color allele. The lady beetles in case 2 display 3 traits. The computer data support that they're determined by 3 different alleles of the wing color gene. These 3 alleles display a dominance order. Then, we access the lady beetle computer model, choose case 3, and set up the model. Students should notice there are 3 traits in the case: broken stripe, normal, and elongated stripe. We then pose the guiding questions. How many alleles do you think are involved in this case? What are their relationships? We can anticipate that some students might say, I don't know. Some might propose there are 3 alleles based on case 2, and some other might bring up the idea of incomplete dominance. We ask students to further articulate their ideas and phrase a hypothesis. The students who propose the 3 alleles may take a hypothetical dominance order and predict what kinds of phenotypical ratios will be observed in the offspring. The students who pose the idea of incomplete dominance may hypothesize a trait as the intermediate trait, then predict the corresponding phenotypical ratios that will be observed in the offspring. From there, we can use the computer model to gather data. Students must test 6 types of crosses because there are 3 traits. Here, I will still demonstrate 5 trials for each cross. You can have your students do more trials. Just keep the number of trials the same for all these crosses. Now we have the data and the patterns. To test the hypothesis of multiple alleles, we will ask students to compare the case 3 data patterns with those in case 2. Students should see the data patterns in these 2 cases are very different. First, the 3-to-1 ratio does not emerge in case 3, suggesting that there's no dominance order. Second, when crossing the beetles with the same trait, we either only get the same trait or a 1-to-2-to-1 ratio in case 3. This ratio does not show in case 2. Therefore, it's evident that the underlying color pattern inheritance model in case 3 is not the 3-allele model in case 2. Then, let's test the hypothesis of incomplete dominance. We bring back the questions. Which trait is the intermediate trait? What is the evidence? When working with students on interpreting data patterns, we may suggest students start with the simplest ones. They will see when crossing two elongated striped beetles, all of the offspring beetles present the elongated stripe trait. When crossing two normal color beetles, all of the offspring beetles present the normal color trait. Since the offspring in the two types of crosses are always the same as the parents, the genotypes of these two traits are likely to be homozygous. We know the normal color trait is determined by Cn. Let's tentatively assign an allele CV to the elongated stripe trait. Then, we can put down CVCV and CNCN for the two traits. Using the Punnett squares, we can well explain the data patterns. When we cross an elongated stripe beetle with a normal color beetle, we only get the broken stripe beetles. Using the Punnett square, we see this cross only generates the genotype of CVCN. If we associate it with the broken stripe, it will perfectly explain the computer results. From there, we can let students predict the results of the cross between two broken striped beetles using Punnett squares and compare the prediction with the actual data. They can see that they match very well. Finally, they can continue analyzing the rest two crosses and we'll see the analysis results are always consistent with the computer data. Now, based on the computer evidence and the reasoning process, students may draw the following conclusions. Two alleles are involved in case 3. The homozygous genotype CVCV determines the trait of elongated stripe. The homozygous genotype CNCN determines the trait of normal color. And the heterozygous genotype CVCN determines the trait of the broken stripe. The broken stripe is the intermediate trait. We can clarify to students that the underlying inheritance model in case 3 is the incomplete dominance model. Because the broken stripe beetles are always heterozygous, you might wonder whether this case is incomplete dominance or codominance. Let's look at the definitions. You can see the key difference is that in the case of incomplete dominance, a heterozygous individual shows a phenotype intermediate in between the phenotypes of homozygous traits. Whereas in the case of codominance, both parents' alleles are fully expressed. When Houston studied these lady beetles, he said, elongated stripe and normal alleles are codominant and produce broken stripe form. Here, he used the term codominant. Meanwhile, Houston pointed out that the offspring beetles, the broken stripe form, do not express both parents' color patterns, but appeared to only display black spots on the wings where both parents displayed black spots. Therefore, this case of lady beetle color pattern inheritance better aligns with the definition of incomplete dominance. We can compare it with the example of human blood types. People with an AB blood type express both A antigen and B antigen. They do not express a trait that's in the middle or overlap. In this lesson, students explore a case in which the intermediate trait is not conspicuous. The case may draw students' attention to analyze data and develop the incomplete dominance model based on evidence. Alright, we have figured out the inheritance in 5 of the 6 forms of common Australian lady beetles. Now you can have your students to figure out the last form, the 5-spotted beetles. You can find the model link below in the description. Let us know what you find out. Leave your comments. Thanks for watching.